Good afternoon, everyone, and welcome to the Helm Video Production Studio product release number 1. This is going to be a quick little talk/demo about, as you can see here, notice my computerized pointer, seeds, what they are, why they're cool, the process of seed germination, and specifically within the process of seed germination, reserve mobilization and what that means and how we are going to study it. But before I launch, let's take a look at a couple of seeds just to sort of illustrate the diversity in seeds. So I think we've seen some of these before. Here, those are broad bean seeds, pretty darn big, and remember they're probably triple that size when they're fully imbibed with water. Here are some lettuce seeds. I'm going to hold it up to the camera for you. Hopefully she'll focus. There we go. Little tiny things, about 1/8 of an inch long. And what else have I got here? I've got some starch containing peas. I have what we're going to work on today. I think I'll put that aside to make an even and smooth segue into our next topic. Zinnias, and I could go on and on and on all day long. So extremely large diversity in size and in form, the largest being something like a coconut. Those are about the biggest seeds around, and the tiniest being orchid seeds, which are microscopic. So all of them have a couple of things in common that I wanted to tell you about. You ready for my computerized transition to a new slide? Wait for it. Here it comes. First of all, let's talk about how they're formed. This is an incredibly detailed botanical drawing of a monocot plant. We have a root system here. We have an aerial crown. We have a set of leaves. This is called a flag leaf. These guys are undergoing photosynthesis. And here we have a little seed, a little maturing seed. It's got a couple of different components that I'll tell you about in just a second. But basically this thing's incorporating macromolecules and the components for macromolecules from two different sources. The first is photosynthesis. All right, light is striking this leaf. It's energizing electrons. The high energy electrons and ATP from those light reactions is fixing this CO2 into sucrose. Sucrose is being transported up the phloem into the seed, and that's making simple sugars and actually polysaccharides like starch that we're going to be looking at today. But that's not the only thing that there is in seeds. There's all kinds of stuff in there. There's proteins, there's nucleic acids, there's trace elements, and all of those inorganic things are obtained from, yep, you guessed it, from the soil. Those are taken up in the root transporters that we've been studying for the last week or so, taken up now in the xylem stream, and then up in the maturing seed, mated with the fixed carbon dioxide that we got from photosynthesis. So the whole seed slowly accumulates these macromolecules. The embryo in the seed, okay, that's going to be the little miniature plant that develops into a seedling when germination occurs. That is developing, so it's a developmental program that's going on. At the same time, the thing is storing a number of different macromolecules. The one we're going to study today is starch, but just know that there's also proteins in there. There's all kinds of things. There's some fatty acids, there's nucleic acids, there's all kinds of things. And at the end of the day, we have a nifty little packet here that has some kind of a protective seed coat, an embryo that's essentially a little miniature plant, and some stored reserves that are going to provide this little embryo the energy and the organic skeletons it needs to develop into a seedling over time. Okay, let me click to the next slide. Okay, here is the subject of today's quote lab, closed quote. We're looking at cereal grains, all right? So let me pull one of these guys out for you to look at, and in particular, come on, you little rascal. In particular, we're looking at wheat, Okay, so there's some wheat seeds, typical monocot cereal grains. Cereal grains are things like corn's a cereal grain, rice, wheat, barley, oats, all that kind of stuff. They're in the grass family. They all have reasonably similar structures, reasonably similar processes that they go through during the germination process. All right, let's see here. I can't sully my workspace. Off. Open my little envelope. Back in there you guys go. And we're good. Okay. So if we take a look at a wheat seed in cross-section, here are the structures that you need to know about. Up here, there's an embryo, incredibly detailed botanical drawing. It's, of course, a living organism. It is a diploid, little teeny plant that's the product of a sexual union during seed development and fertilization and maturation on the mother plant. Okay. There's, in monocots, this dead triploid endosperm tissue, and that's where all of the stored reserves are. So we've got starch in there. We have things like ions, trace minerals, stuff like that. And we have proteins, and some of you might be interested to know that the major form of protein in wheat are glutens. So the gluten proteins provide the young embryo as it's germinating and growing into a seedling with amino acids that can then be formed into new proteins that the thing needs as it grows. And for us as people who like bread, usually, the glutens are what provide the sort of the stretchiness in bread dough and give things like a loaf of bread its nice chewy texture. And also it makes it nutritious as well. Okay, so you take this wheat seed here, or any seed for that matter, and you provide it with all of the requirements in terms of water and environmental conditions and whatnot. Let some time pass, and a germination process ensues. Okay, what is germination? Well, germination involves a lot of things that you guys read about, and I know you did it because I saw your quizzes. There's the uptake of water. There's the resumption of metabolism. There's starch, proteins, all sorts of other sorts of things in there, and all of that stuff is getting mobilized. In the meantime, mitochondrial reform, transcription and translation begin almost immediately. As a matter of fact, you can detect respiration, mitochondrial respiration in an imbibing seed within like 15 minutes of when you add water to it. So it comes to life, so to speak. It wasn't dead in the first place, but it becomes metabolically active very, very quickly upon imbibition. So on we go. The process of germination actually is considered to be complete when the young little root, which is termed the radicle, protrudes from the seed coat. I'm going to check ahead here. Here's a little seedling. It is still underground. All right. It's got a couple of little structures. Here is, I've got to back up and tell you something in a minute. Here is a little structure called the coleoptile, which is going to protect the young leaves from gravels and mechanical damage as the thing punches up through the soil. This is the root system. Since it's a monocot, it's got a fibrous root system. Oh, yeah, that's right. We have our starchy endosperm. The third cell type that I wanted to tell you about is the alurone layer. The alurone layer. The alurone layer is a living cell layer that surrounds the endosperm, and it actually has a pretty critical role in the germination process. It is secretory tissue, okay? That means that in response to some external stimulus, the cells in the alurone layer can make any number of different so-called hydrolytic enzymes or hydrolases that are secreted through the endomembrane system, the ER and the Golgi and whatnot, into this dead starchy endosperm, and those hydrolases then cut the macromolecules from their polymeric form into simple subunits, such as simple sugars like glucose. Those simple subunits are then absorbed by a specialized structure here called the scutellum. That's part of the embryo. And it's sort of like, if you want to think of it analogously, it's sort of like a placenta in a vertebrate fetus. All right, so it's an absorptive tissue, and its major job is to take up subunits that are released from the starchy endosperm as the hydrolysis of those stored reserves takes place. So after a pretty short time, you go from this quiescent, non-metabolizing dry seed into a young seedling. That's the basic process of germination, and those are the tissues that we're interested in today. I'm going to do something here first. I'm going to start so you don't have nothing to look at. I'm going to put some seeds down that I have had in the soup. Turn it right side up for you. And there's a little time course. Hold on just a second because I'm going to show you something. See, my little video productions have demos in them. Got a couple of things to show you. Okay, so this is seeds that I imbibed in a refrigerator overnight. These are seeds that have been imbibing for 24 hours, and these are 72 hours right here. Okay, so these are all wheat seeds. And let's take a look at this. Even though they were imbibed in the refrigerator, you can see, I don't have the dry ones handy, it's pretty obvious. I don't think this is going to focus too well. I need you know what I've learned is you need a straight line. Let's see if this works. Maybe yes. Yes, that'll work. My little camera focuses on a straight line. Come on. Shucks. Anyway. This are seeds that have been imbibed for just 24 hours. And I'm going to again see if I can get these guys to focus. Come on, guys. Shucks. Let's try that. Okay, 24 hours. I'm going to be one step ahead of you, little rascal. Let's take a look at what's happening after only 72 hours. You can see it's still not focusing super well. Not bad though. So you can see there's all sorts of action. Get a dissecting needle here. I want to show you what this actually looks like. That's the little coleoptile right there, and then these are the little roots. Let's pick it around a little so you can see it. Hope you can see that all right. Okay, so that's what those guys look like. Now, what I wanted to show you was a little demo that I've got, and for that I'm going to move the Helm Production Studio to what I call the micro mode. Hold on just a second. Bear with me. I'm going to move the camera like that. Yeah, what do you know? Miracles happen from time to time. Let's get our light adjusted. All right, and then I'm going to boost this dude up a little bit. Something I want to show you. Here, we have our 24-hour seeds. Let's see. Now that that seems to be working well, let me show you the 72s again. Take another look at 72 hours. They're kind of cool. There we go. Yeah, excellent. Okay, I want to take one of these 24 hours that I have cut in two, and I'm going to stain it with some Lugol's iodine solution. Now, Lugol's iodine solution is an iodine-based solution. I wonder why they call it that. Okay. There we go. Put this guy up here so you can see it. Let it focus for a second. Come on, you little rascal. You can do this. I know you can. There we go. Got it. All right, beautiful. Okay. The autofocusing is an unforeseen setback. All right, so I'm going to take a drop of this iodine solution, and Lugol's iodine stands for starch, so I'm going to get just a little drop. See, I've schlepped Lugol solution. By the way, you can see the paper towel changing color. I've noticed, glad I dry run this thing. I've rehearsed it a little bit anyway. Lugol's also stains cellulose. Look at that. Completely black on the inside. So these little seeds are absolutely packed full of starch. Oops, I don't think I wanted to throw away. Yeah, that's fine. All right. So those are the tissues. Now, once the process of reserve mobilization has taken place, imbibition's taken place, and the thing is fully metabolically active, let's go back up here now. Okay. We get, how do you like that? A little seedling. All right, so the seedling here is now a couple of things. First of all, notice that we've developed a pretty extensive root system. Here, this white thing that I've indicated is the coleoptile itself. Let's see, you can see it a little better. And there are the young leaves that have come out through the protective coleoptile. So once this thing comes up through the surface of the soil, the coleoptile is able to perceive light. It stops elongating, and out pop these little leaves, and you're ready to rock and roll. So there you go. We have a nice little seedling that is now growing and happy and having a grand old time. So, I think I was supposed to show you something here, but I don't remember what it was. I think I've already done it. Today, we are going to study alpha amylase. So we're going to study a particular prominent enzyme that's easy to study. It's a good model enzyme in the study of reserve mobilization in seeds. So yes, the name, as you know, is alpha amylase. Alpha amylase reaction, which I think you all got right, is simply the cleavage of alpha-1,4 glycosidic bonds. So it takes starch, which, as you know, is an alpha-1,4 glucan, and it breaks it down, snipping one at a time, alpha-1,4 glycosidic bonds to release glucose. And then the glucose then is used by the embryo for two things. It's used for mitochondrial respiration, and it's the glucose, the sugar molecules themselves, are also used for the creation of new macromolecules that little seedling's going to use to grow. And remember, when it's underground, it can't do photosynthesis, so it has to rely on these stored reserves before it has a developed root system, and B, leaves from which it can do photosynthesis. So we're looking at alpha amylase, and we're going to do a couple of different little experiments. We're going to look at the timing of alpha amylase accumulation and the timing of alpha amylase activity induction. We're going to look at the location of the enzyme itself in the seed. We're going to look at the signaling and the regulation of it, and that's, to me, kind of the most interesting part of this whole story. We're going to look at a hormone called gibberellic acid, or GA, but we're also going to look kind of carefully at where that hormone is originating from and which tissues are perceiving. Amylase is what we're going to study here. We're going to do a couple of different things. This is where I'll show you some stuff. After this, we're going to take seeds that have been treated in various different ways. I'm just going to hit my space bar every couple of minutes. We're going to take tissue that's been treated in various different ways, imbibed for different periods of time. I'm going to bend this just a little teeny bit this way. That's good. There we go. So we're going to take tissues that have been treated for different conditions and for different periods of time. We're going to make native extracts from those. In other words, we're going to take the tissue, we're going to add some kind of a buffer. It's basically a low pH buffer. It's got no detergent in it. All it does is release cytosolic contents without denaturing the proteins. That's what native means. So the proteins themselves are still going to be in their active form. And from and using those native extracts, we're going to do two different things. We being in quotation marks. We're going to virtually do two different things. First, we're going to take little samples of these extracts, and we are going to treat them with SDS, which is an ionic detergent, with beta-mercaptoethanol, which reduces disulfide bonds and breaks them apart. We're going to boil those now denatured extracts, and we're going to load them onto sodium dodecyl sulfate gels, polyacrylamide gel electrophoresis, and we're going to do SDS page. So we're going to take our little protein samples and we're going to load them into wells on these gels, and we are going to apply an electrical field, and the proteins are going to migrate through the gel according to their molecular weights, with smaller proteins getting an easier time through the gel matrix and going faster, and the larger proteins having a harder time wiggling through the gel matrix and therefore going slower. Since the proteins themselves are all denatured, we're only really separating on the basis of size. Okay, we're not separating on the basis of charge or shape. So the proteins are fully denatured and they're coated with little negative charges, so the charge to mass ratio is the same in all of the proteins. So the only thing really determining their migration rate through this polyacrylamide gel is going to be the molecular mass of the protein itself. Okay, so that's number one thing. When we're done, we're going to stain our gels with Kumasi Brilliant Blue. And Kumasi Brilliant Blue is a general protein stain, and it stains all proteins blue color, and the intensity of the band that you see on one of these gels reflects the amount of that protein in the gel. All right, so that's SDS page. And I've got a picture of an SDS gel that I'm going to put up online for you, okay? The other thing that we're going to do is we're going to take some of these native extracts and we're going to do enzyme assays. I'm going to do enzyme assays, and I think I've told you before. I'll just tell you about it. So we're going to take some extract, we're going to put it in a tube with some starch, we're going to time the sucker. I'm going to put this guy up. So we're going to get an assay, we're going to time it, we're going to put equal amounts of extract in each tube, and we're going to add starch, and we're going to measure the disappearance of starch during our assay period. All right, and we're going to measure the amount of starch with an iodine stain, and just pop it in a spectrophotometer and read the absorbance. Now let's think about this a little bit. Here, enzyme or assays that contain enzymes with higher activity are going to chew up more starch. So let's think back. We did an experiment with nitrate reductase a few weeks ago, and you might remember with nitrate reductase we were looking at the accumulation of product. Remember, we were doing nitrite assays, and the greater the activity, the greater the rate of nitrite accumulation. Here, we're doing the disappearance of substrate. So that's the other basic way that you can measure the activity of an enzyme. So here, we're looking at starch disappearance. We're going to measure starch by iodine staining, and extracts that have a higher activity then are going to chew up more starch, and they're going to have a lower absorbance. All right. Now what you're going to have to do, based on what you learned before with nitrate reductase, is you are going to have to find the activity. Okay, you're going to have to determine the activity. So when you hand in your lab exercises, you are not going to graph just absorbance, because that'll give you what in the scientific world we call Bass-Ackwards results. You're going to want to express this as activity. All right. And I'll put a little sheet up online like I did before to help you figure out how to determine activity. So we are going to figure out in these assays how much starch is used per minute per milliliter of extract. So that's the units you're going to use. All right, so we're going to do enzyme assays. So that's that part. Let's go back to this. So we're going to do SDS gels and we're going to do enzyme assays. What are our treatments? Let's take a peek at that. Look at that. I'm going to move this, and I think you can see this. And I'm going to go through it with you. These are the basic treatments that we're going to be using. First, we're going to do a time course. All right, so this first 4 treatments here represent a time course of induction. And you can see that there's 0, 24, 48, 72-hour whole seeds. All right, so that's one set of treatments. The next 2 we're going to do, or we did do, or maybe someday you will do, we're going to do, and by the way, this is on page 66 of your lab book, we're going to do some dissections. All right, so we're going to take these 72-hour imbibed embryos, did it. What do you know? So if we were real and doing this for sure, we would just dissect away the embryo portion and we would dissect away, I guess I would say, the endosperm and alurone. Okay, so we're going to actually do a dissection. What we're going to do is you would be given some seeds, all right, whole seeds, and you would cut them here. So you would have an embryo, and then you would have the dissected away endosperm and alurone. So that's what those 2 treatments are. Okay, so that's at 72 hours, and they've been dissected. Now these bottom treatments, I guess I'll just leave it flat here for you. You can follow along, but these guys here are 48 hours. Okay, so these are 48 hours. And in those, what we would have done is before imbibing these things with water, we would have cut the seeds in half. All right, and there's 4 basic treatments here. First, 48 hours cut in half. Reading this thing upside down. This is the embryo 1/2 seeds. Okay, so that's the half seed containing the embryo, and the endosperm 1/2 seeds. I'll show you what that means. With that, what we would do before I imbibe them, I cut them like this, and I'll even show them to you in a second. But I took dry cut seeds and I imbibed these for 48 hours, and I imbibed these halves for 48 hours. And I did 2 things. First, I just imbibed them in water, and then a duplicate set I imbibed in the presence of a physiological concentration of Ga. All right, so just kind of think about that for a second. The embryo and endosperm dissection, the entire endosperm half is in one set of extracts, and then the embryo alone is in the other set of extracts. And then these half seeds, the embryo 1/2 has embryo, alurone, and endosperm. The endosperm 1/2 has alurone and endosperm only, but no embryo. Okay? So that's kind of an important distinction for you. There's one additional treatment that I wanted to tell you about, and that is 48-hour whole seeds imbibed with cyclohexamide. You have to scratch your little noodles and look up what cyclohexamide does, and the reason I'm not telling you is that we did it before when we worked with nitrate reductase. So we've done that before. All right. So that's the basic drill here. So remember, we're going to take some extracts, we're going to do enzyme assays on this, and then we're also going to run an SDS gel on this. And the lane assignments for the SDS gel are given on page 68. Let's see. Page 68 of your book. All right. So what do I got here? Then, after you chew on your data, you're going to have great revelations. And one other thing I wanted to show you is just a whole bunch of germinated seeds that I just happen to have. You go to the Honest Weight Food Co-op, you can actually get seeds for sprouting. Okay, so you can make sprouts that you put on salads and sandwiches and stuff, and it's just a mix of seeds. And I want to show you what happens after about 5 days. Okay, check this out. Amazing, huh? So these guys are almost ready, just to give you a sense of scale. Here's my hand, so I got this little sprouter at Honest Weight. Just a little jar with a lid. You put some seeds in here, and all of those processes that I told you about are occurring in here, and within the next day or two I'm going to have some real nice stuff that we can put on salads and sandwiches and whatnot. There's one other nice application to seed germination, and in particular to alpha amylase, that I wanted to tell you about, and that is the creation of beer. So I wanted to tell you a little bit about the creation of beer before I end this video. I've put actually a recipe up for you, and I wanted to tell you also that I'm going to start an all-grain beer this weekend. So it's one of my little hobbies that I've got, and it's pretty cool and it's pretty fun. So I thought you might be interested, particularly Matt. Matt might be interested. I know he worries about his liver. Anyway, I wanted to tell you a little bit about how beer is made. So if you're ever bored at a party and read the back of a Coors bottle, you will read that the beer contains malted barley, and it contains hops, and it contains yeast, and it contains Rocky Mountain spring water. Okay, so what do all those things do? Well, malted barley is barley that's been germinated and then stopped mid-germination, and in many cases roasted. Okay, so unless you roast it, the dried down malted barley actually has active alpha amylase in it. So what you do is you take malted barley and you add hot water, and then the hot water, not boiling water mind you, but hot water. In the hot water, the alpha amylase, am I going to do this right? No, that's not right. There we go. Not bad for upside down, huh? The alpha amylase breaks the starch down into simple sugars during the mashing process, and this is about 150 degrees Fahrenheit or so, so hot, but not boiling. When you're done with that, you have this weird stuff called wort. So you get rid of the old spent grains, you get rid of the husks, you get rid of all that stuff, and you're left with this really sweet, kind of syrupy, brownish liquid. And what you do is you boil that, and at some points, various points in there, you add hops. So hops are little flowers that come off of a vine, and they have in them alkaloids that are bittering agents, and they also have antimicrobial properties, so it's kind of cool. So you boil your wort, typically for an hour or two. When the boil is done, you let the whole works cool off, and you put it in a container, and you pitch some yeast. And the yeast then metabolize the sugars that were released from the starch by alpha amylase. And since you do this in an airtight container that allows CO2 to exit but no oxygen to get in, the yeast is in an anaerobic mode and performs fermentation. And the products of fermentation, as you probably know, are carbon dioxide and ethanol. So after you let the fermentation go to its total conclusion, you then take your beer, and typically what you do is you add just a little bit more sugar right before you bottle it, and that allows a teeny bit little more fermentation, which gives the beer its fizziness. Okay, so I better put a cap on here, huh? So you put it aside for a couple of weeks, and then you put it in the fridge, and then you pour it into a glass and enjoy it. So that is how you make beer. So I've put a recipe for an all-grain beer up there. If you ever want to try it, I would not recommend doing an all-grain beer for your first beer, but there's a whole bunch of pretty easy kits There's malt extracts that you can buy. There's all sorts of different varieties of hops that you can buy. There's all sorts of yeasts. As a matter of fact, there's a place called Homebrew Emporium over in East Greenbush where you can buy all of this stuff. And it really is pretty fun, and the beer you get is actually quite good. And I'll be starting some this weekend. So that is the story with alpha amylase. The processes of seed germination, imbibition, reserve mobilization, and how those processes, at least to some extent, are controlled by plant hormones such as gibberellic acid. So I'll have some data up for you in a little while. I hope this has helped you to understand what you read in the lab book and in the textbook, and I hope that you find the exercise interesting. So enjoy, and I'll talk to you soon. Take care. Signing off for now.